Hey everyone, and welcome. So let's get right into it. We're going to tackle one of the biggest questions out there. How does life actually work? You know, how does a tiny little seed have all the information it needs to grow into a massive tree? Or how did you become, well, you? It turns out the answer is inside this incredible biological instruction manual. And today, we are going to figure out how to read it.
I mean, really think about that for a second. Inside every single one of your trillions of cells, there's a master plan, a complete blueprint with all the instructions to build and run your entire body. So, what is it?
Yep, it's this amazing molecule, deoxyribonucleic acid. We just call it DNA. And you can make a pretty good argument that it's the most important molecule we know of. It is absolutely the star of our show today. So, here's our game plan. We're going to start with the blueprint itself, what DNA actually is. Then we'll look at how its incredible structure was figured out. After that, we'll see how it makes perfect copies of itself, how that code gets turned into a usable message, and finally, how that message is used to build, well, everything. It's the most amazing information system in the universe.
All right, first up, every story's got a beginning. And for us, that's the blueprint itself. Before we get into how it's copied and read, we've got to understand what this stuff is actually made of.
Okay, so the official definition calls it a long polymer. Now, don't let the fancy words fool you. That just means it's a super long chain made of repeating parts, kind of like beads on a string. These repeating parts are called nucleotides. And for pretty much all life on Earth, this molecule is the hard drive. It's the stable, reliable place where all the genetic information gets stored for the long haul.
So, for a long time, scientists knew DNA existed, but they had absolutely no clue what it looked like. Figuring out its shape was one of the biggest, most exciting scientific races of the 20th century.
And this is what they found, the legendary double helix.
Back in 1953, James Watson and Francis Crick put forward this beautiful twisting ladder idea. Now, their model was a work of genius for sure, but it absolutely depended on the incredible X-ray work done by the chemist Rosalind Franklin. And what they all discovered was that this shape, it wasn't just elegant. The structure itself held the secret to how life could copy and pass on its instruction.
Let's stick with that ladder analogy. The two long sides of the ladder are what we call the backbones, and they're made of sugar and phosphate. You'll notice they actually run in opposite directions. That's what anti-parallel means. But the rungs of the ladder, that's where the magic really happens. The rungs are made of 4 chemical bases: adenine, guanine, cytosine, and thymine. Let's just call them A, G, C, and T. And here is the golden rule, the rule that makes everything else possible. A on one side always hooks up with T on the other, and G always hooks up with C. That's it. That simple rule is the foundation for all of genetics. And this is where the pure genius of the design comes in. Because of that super strict A to T, G to C pairing rule, the two strands are perfect complements to each other. So if you know the sequence of letters on one side, say A, G, G, T, C, you automatically know the other side has to be T, C, C, A, G. It's like having a perfect photographic negative. The structure itself is practically screaming, "Hey, you can copy me!" Okay, so the structure is basically begging to be copied. But how does that actually work? I mean, how does a cell make a flawless copy of a 3 billion letter instruction manual every single time it needs to divide? Well, Watson and Crick had a hunch right away, and it was this beautifully simple idea called the semi-conservative model. The ladder just unzips right down the middle. Then each of the old strands acts as a perfect template to build a brand new partner strand. And what do you get in the end? 2 identical DNA molecules, each one is a perfect hybrid, half old and half brand new.
Now, this was a great theory, but in science, you've got to prove it. And they did, with this incredibly clever experiment. Scientists grew bacteria using a heavy type of nitrogen, so all its DNA was heavy. Then they moved the bacteria to a medium with only light nitrogen. After one round of division, they checked the DNA. And guess what? It wasn't heavy and it wasn't light. It was exactly in the middle, a perfect hybrid. That was the proof. Each new DNA kept one old heavy strand and built one new light one. Nailed. Okay, so we've got our master blueprint and we know how to copy the whole thing. But think about it. If you're an architect, you don't take your one-of-a-kind original blueprint to the muddy, chaotic construction site, right? No way. You make a photocopy, a disposable, working copy. And in the cell, that's where our friend RNA comes into the picture. And the process of making that working copy has a name. It's called transcription.
A little segment of the DNA unwinds temporarily, and one of the strands is used as a template to build a short, single-stranded molecule of RNA. This is the very first step to actually using the information stored in the DNA. So, what's the difference between the DNA blueprint and the RNA message? Well, there are three main giveaways. First, the sugar in RNA's backbone is slightly different. Second, and this is a big one, RNA doesn't use the base thymine, T. Instead, it swaps in a different base called uracil, or U. So in the world of RNA, A pairs up with U. And third, while DNA is that stable double helix, RNA is just a flexible single strand. It's built to be a temporary, go-anywhere message. All right, we have our message. The RNA photocopy is ready. Now it's time to take that message out of the main office, the nucleus, and head out to the factory floor of the cell to build the final product. And that product is a protein. This whole flow of information is so fundamental, so core to how all life works, that Francis Crick gave it this really grand name, the central dogma. Information is stored long-term in DNA, it's copied into a temporary RNA message, and then that message is translated to build a protein. DNA to RNA to protein. That's the basic logic of life right there. Now, building a protein takes a whole team. You've got the messenger RNA, or mRNA, that's the work order, the instructions. Then you've got the ribosome, that's the factory itself, the machine that reads the instructions. And then you have the real genius of the system, the transfer RNA, or tRNA. These are the specialized workers that can actually read the message and go grab the exact right building blocks. And this is that amazing little adapter molecule. Seriously, think of it like a universal translator. On one end, it has a 3-letter code that plugs perfectly into a matching code on the messenger RNA. And on the other end, it carries a very specific building block, an amino acid. This incredible little molecule is the physical bridge that connects the language of genes to the language of proteins. It's brilliant. So what language is this code written in? It's a language made of 3-letter words called codons. If you look at this chart, you can see how it works. For example, if the factory reads the word GUA on the message, a tRNA brings over the amino acid valine. If it reads UCU, it brings serine. See? With 4 letters to choose from, arranged in words of 3, you get 64 possible combinations, which is more than enough to specify the 20 different amino acids that are used to build all life. And how does the factory know where to start and stop? Simple. Just like a sentence, the message has punctuation. There's a special start word, AUG. The ribosome latches on there and just starts reading, word by word, until it hits one of the 3 stop words. This makes sure the protein gets built with the perfect beginning, middle, and end. And voila! The final result is a long chain of amino acids, which then folds up into a beautiful, complex 3D shape. And that folded chain is a protein. These are the little machines that do practically everything in your body. They digest your food, fight off viruses, give your cells structure. That seed becoming a tree, that's proteins doing all the work. The instructions from the DNA have finally become action. I mean, it is just an astonishingly elegant system, right? And it happens trillions of times a day inside of us with incredible accuracy. But that brings up one last, really big question. If this system is so perfect, what happens when it's not? What happens when there's just one little typo in that 3 billion letter code? And in the answer to that question, well, that's where you find the secrets to everything from genetic diseases to the entire engine of evolution.